Welcome, everybody. My name is Jaxton Willman. I'm an undergrad researcher from the University of Florida in the Saxena Lab for Neural Control. Today I'll be talking about our latest work developing a neuromusculoskeletal model including neural activation, muscle contraction, and skeletal dynamics for targeted sensorimotor control. This work takes a controls theory perspective on sensorimotor control. From previous research, we understand that the computational role of the motor regions of the brain are to drive a complex, multidimensional arm to achieve a specific goal, for example, reaching from point A to point B. Thus, we want to understand the neural signals better by understanding how they control our limbs. This can help us understand how to fix things when they break down, in the case of motor diseases and disorders. We have two important goals for our work. The first is to understand the control strategies implemented by the brain to drive movements, and our second goal is to improve assistive technologies for motor diseases and disorders, including neurodegenerative diseases that affect movements. To this extent, we are developing a brain-inspired closed-loop controller for an anatomically and physiologically accurate arm model. In her previous work, my research advisor Shreya Saxena looked at a neurally inspired controller to drive a musculoskeletal model. My focus today is on the musculoskeletal dynamics, section C bordered in green. She used a single joint model under the class of equilibrium point models for motor control from McIntyre and Busy. However, in her case, the musculoskeletal model was linear and not multidimensional. What happens in the more realistic case of a biomechanically accurate arm model? Let's find out. Previous research in this field details the internal representation of the sensorimotor loop within the CNS, as shown in this figure by Wolpert et al. The analysis of behavior has led us to an understanding that driving a limb model can be performed by a combination of inverse and forward control models. However, the neural basis of these models is not clear. As research focusing on motor control, our work explores a possible computational model of the sensorimotor loop. Another research piece we relied on was by Wellington Casio-Pinheiro et al., who performed system identification using OpenSim dynamics, but for a two-muscle, one-joint human arm model. They used a recursive instrumental variable method for system identification of the musculoskeletal dynamics. We will build upon their work and use models which are more biologically accurate. As an overview of the methodology, we start by generating motions. Then we use the OpenSim CMC tool to get muscle activations. We perform system identification and then we analyze the NRMSE values. For our arm model, we are using a computational 6-muscle 2-joint human arm model created in the OpenSim software. OpenSim, created by Scott Delp et al., is a wonderful software that users can develop models of musculoskeletal structures and create dynamic simulations of movement. OpenSim provides a computational muscle control or CMC tool that takes a defined motion and generates the needed muscle activations to achieve it. So first we need to generate motions. The motion file is an XML file specifying an ideal motion described by the changes in the elbow and shoulder joint angle with time. Our described movement is 1.1 seconds long, with the first 0.1 seconds defined as zero movement with an elbow joint position of 30 degrees, which acts as an initial condition. This initial condition allows analysis of the muscle activations from a baseline. We pseudo-randomly generate 5,000 motion files for the training data and 500 for the validation data. Next, we use OpenSim CMC tool to generate the corresponding muscle activations to achieve our specified motion. While generating the muscle activations, the tool uses them at each time point to compute the resulting positions and speeds of the elbow and shoulder joint and the muscle fiber lengths. We run this on all 5,500 motion files. We begin by compiling the resulting muscle activations as the inputs and the states of the joints as the outputs into an ID data file ready for system identification in MATLAB. We use the N4SID function in the system identification toolbox in MATLAB to generate discrete time state space models with a time step of 0.001 seconds ranging from orders of 8 to 15 to determine which system order is more ideal. We compare the systems against the training data and the validation data, and then compute the average normalized root mean squared error, or NRMSE value, across all experiments per system order as a measure of system performance. This is a graph of the ideal motion and the resulting motion from the CMC tool. The first and last 0.03 seconds are chopped off by the CMC tool. There is a very slight difference only near the vertex of the concavities. Presented here are the first 500 training data motions. The graph on the left shows the ideal elbow position from the defined motion file over the 1.1 second period, and the graph on the right shows the resulting elbow position states after running the motion through the CMC tool. This graph displays the training data system response for each system order we computed. This is a graph of the first motion of the training data. The muscle activations are the input, and the position and speed of the shoulder and elbow joints are the outputs of the system. The legend displays the NRMSE values for each system order. While this is an example of a good simulated response as the accuracy is high, there's a different ideal system order for each output. The system represents some experiments very well with an average NRMSE of around 90%. This graph displays the system response on the validation data for each system order we computed. This is an example of a bad simulated response as no order can accurately determine the elbow value for the first motion of the validation data, as all NRMSE values are above 1000%. The model is not performing well on held out data yet. OpenSim's neuromusculoskeletal dynamics are nonlinear, so there will be loss when trying to approximate the dynamics with a linear system. While the systems are doing well on some experiments with an NRMSE of about 90%, it does not yet have a good average NRMSE on training data, perhaps due to the large diversity in the simulated motion. With 8 inputs and 4 outputs, there is a very large parameter space we are trying to simulate, and so only 5,000 training simulations may not be enough to learn the parameters of the system. In ongoing work, we are developing more accurate linear dynamical systems for better signal reconstruction. Our next milestone is to design a neurally inspired controller to drive the linearized musculoskeletal dynamics to a desired position. This framework can be used in the study of assistive neurotechnologies for combating the effects of neurodegenerative diseases, for example, by designing compensators to reduce the effect of weaker neural signals to the muscles. Thank you very much for your attention.